Hi. So today my presentation has to do with an article called Targeting Inhibitory Cerebellar Circuitry to Alleviate Behavioral Deficits in a Mouse Model for Studying Idiopathic Autism. The study was conducted by Chao et al. And published in March of this year. So to start off with uh, this presentation, I'm going to explain autism spectrum disorder and the cerebellum. Um, in short order to help the whole thing go forward more smoothly. So let's see how it works out. So you may have heard of this disorder. Autism spectrum disorder is a developmental disorder that may influence many different mechanisms within an individual's nervous system, such as irregularities or problems with sensory processing. You may see impaired understanding or detection of nonverbal communication cues, or you may see varying deficits in social or occupational functioning and intelligence scores, such that a wide spectrum of individually unique configurations manifest across the population. However, for the purposes of this study, we're going to look at mostly the behavioral and social deficits. So in humans, you may see something called stimming. It's a ritualistic or compulsive seeming self-stimulation activity where someone may move a limb back and forth. They might bob um, back and forth. They might wiggle around, or they might spin. It's usually in response to some sort of sensory overload or some sort of social overload. You see restricted patterns of behavior. Moving on, this also has to do with difficulty adapting to changes in or transitioning to a new activity. Problems with flexibility, I guess. They depend on routine and sameness all the time. You see impaired fine motor learning, or both gross and fine motor skills are actually affected, but fine motor learning seems to be particularly affected. In relation to this, there's also this interesting tidbit where they have less brain hemispheric asymmetry than the regular population. Okay, so moving on, you'll see a situational lack of social inhibition or monitoring during social contexts. You'll see difficulty adapting to changes in or transitioning to new activities. So we covered this before, but um, if they expect a certain activity during a certain part of the time of the day, they might be inclined to do it no matter what. And you see them interact sort of in a very high variance way to language. You see individuals with more classical autism um, use more rudimentary versions of it or alternate versions of language or vocalization or noise through the mouth, basically, to communicate different things. And you also have expert level people who might be um, high level experts within a certain topic or subject. So they might have a lot of linguistic flexibility and expertise or skill or dexterity. But how this study has to do with the mouse model for autism, which is centered around the BTBR mice compared against the WT species. So they have reduced sociability. This certainly has a counterpart in the social monitoring um, thing in humans. You also have ultrasonic vocalizations, which corresponds to language activities, and you have increased self-grooming behavior, which corresponds with stimming. And you have psychiatric comorbidity of anxiety and depression with autism spectrum disorders. The authors mentioned this, and anatomical similarities with ASD patients that rats may have or mice may have. So to move on to the cerebellum, We have, remember, the thing that we're supposed to remember about the cerebellum, no matter what we remember about this class. It has roles in regulating posture and balance, coordination of skilled movements, and muscle tone. The structure is, however, a region of interest when we're talking about autism, where there's like a lower number of Purkinje neurons, there's a lower vermal volume, lower gray matter, lower mass, decreased cerebellar activation during cognitive and social processing, and disrupted cerebrocerebellar functional connectivity. Purkinje neurons get specific changes apparently due to genetic profile results um, from having ASD or um, the ASD equivalent within a mouse. So you see lower excitability for Purkinje neurons. It takes more to get them to fire, and um, this is due to GABA release from interneurons. And this has more to do with the hypotheses that we're going to see generated on the next slide. So for introduction, uh, here they basically talk about, besides whatever I just went over, about how there's a lot of genes involved, but they mostly involve overlap with other disorders, so it's hard to tell a lot of things. And there's also a lot of difference in symptoms. Person A might have symptom X, person Y may not have symptom um, X, they may have symptom B instead. So you see a lot of varying degrees of severity. So that's why you see it being called a spectrum. So you see classical autism on one side and high functioning autism, or Asperger syndrome as it was called in the last decade or so. And through the use of animal models, we can basically go on to access certain answers about autism without using humans, which I'm sure you can tell would cause a lot of ethical headaches. But this information allows us to potentially create pharmacological treatments down the line for humans if everything works out properly and things are viable. And the technology is there and everything falls into place. Anyways, remember, um, for autism, the general theory seems to have the consensus that there's reduced pruning of synapses during development. So, an autistic individual might have more synapses left over from their time in development as opposed to a non-autistic person or a neurotypical person. So such individuals may also exhibit different patterns of activity in their default mode networks. So hypothesis 1, BTBR Purkinje neuron firing rates in those rats, BTBR rats, is going to be lower than that of WG counterparts. Hypothesis 2, after nullifying excitatory and inhibitory inhibitories synaptic inputs, Purkinje firing rate decrease can be explained by amplified GABA release from interneurons, which also results in lowered excitability of Purkinje neurons. Hypothesis 3, a low threshold potassium channel in the axonal terminal of interneurons is downregulated. DHA, the Kv1.2 antagonist, Kv1.2 is the name of the potassium channel. DHA seems to counter the inhibitory overtone and the social deficits of BTBR animals in vitro and in vivo, respectively. So this sort of helps to counter Purkinje neuron firing rate being low, and it helps regulation kick in better and more efficiently. And then we have hypothesis 4, which sort of stacks with with the thing from hypothesis 3, and they would work together if they both turned out to be true. So to move on, hold on. Okay, there we go. So um, BTBR and WT mice were used, mostly male, as per the autistic population. Brain dissection was used sagittally. They used multiple age groups. They blocked AMPA and NMDA receptors to block GABA and also blocked GABA receptors in different conditions. They used like electrodes and stuff to record excitatory postsynaptic currents. Um, they took care of antibody requirements and they blocked certain membranes. They applied DHA through a membrane in the abdominal area um, and also on the cerebellum. And they also used intracranial injection on the cerebellum of younger BTBR rats, whereas older counterparts went through stereotactic surgery for those viral vectors. Um, so they used cell-attached patch clamp recordings for multiple parts of the experiment on multiple conditions. So they had to use, by way of statistical analyses, ANOVAs. They used between subject factors for groups, within subject factors for conditions, objects, and intervals, repeated with two-way ANOVAs with within subject factors for treatment, condition, object, and interval. Additionally, they did post hoc tests after detection of significances, so that you have it. And the behavioral tests that they did were different in that they mostly tested social function with the three-chamber social test. They tested avoidance responses because rats or mice are afraid of open space with lots of light. So they saw how long they were stuck somewhere and didn't want to move away from it or so on. And they had an open field, which was generally just um, the open field task had physical activity, how much they moved, how much they ran, moved somewhere, how long, how little time they spent stuck in a dirty, dangerous, openly lit space. So it turns out that all four hypotheses that were stated in the last paragraph of the introduction turned out to be validated by the results. And we see increased GABA release from interneurons. They do affect Purkinje neuron excitability. There is reduced output from the cerebellar cortex level where you see lots of Purkinje neurons. There's poor precision and lower regularity with Purkinje neurons of BTBR rats as opposed to WT rats. Purkinje neurons normally signal using GABA to deep nuclei in the cerebellum to help manage and modulate or moderate cerebellocortical thalamic connectivity. But within autistic individuals, that sort of regulation doesn't happen. So you might see less regulation. You might even see problems with executive dysfunction down the line, downstream from that. You never even know. Um, so uh, the KV 1.2 channel and agonist improves social affective function. So social and emotional function are improved by countering presynaptic inhibition. So Purkinje neurons keep firing more, and also in the you by using the other chemical, they also saw that chemogenetically, like chemically, stimulating the membrane of Purkinje neurons can also affect BTBR mice from exhibiting more autistic behavior. So it might make them behave more like the average population of rats. So to move on to the discussion, they talked about contrasting like their findings with how in in the hippocampal area for BTBR mice, there's actually more activity. Um, like the ratio is like the reverse of whatever we have with the cerebellum. However, here we have with the interneuron GABA release and KV 1.2 channel function. Um, we can see how Purkinje neuron membranes serve as potential precursors to future pharmacological treatments, and we can use these findings further down the line and exploit this mechanism or this doorway to find new answers to problems. So they also discuss limitations about how these mice have no corpus callosums or how their cerebellums may have abnormalities that may not apply to mice as a whole or so, or DHA and CNO applications. They talked about how, like, when the whole cerebellum was involved, as opposed to just the posterior cerebellum, the rats seem to show um, a lot of non-social benefits as well. So, yeah, the cerebellum seems to be involved in a lot of different things. So, downstream disinhibition of deep cerebellar nuclei may gate thalamocortical information processing, and that may affect like a um, larger number of networks downstream or further downstream um, from what they discussed within the discussion part of this paper. Anyways, moving on to content questions. Which single main type of neuron in the cerebellum does the article deal with? Purkinje neurons. What traits in BTBR mice correspond to models of ASD? Altered ultrasonic vocalization, increased grooming behavior, and reduced sociability. What neurotransmitter does the Purkinje neuron use to signal to deep cerebellar nuclei? GABA, or GABA aminobutyric acid. Okay. What did the behavioral tests measure? Sociability, motor activity, and avoidance responses. How does the firing rate of Purkinje neurons in BTBR mice get decreased? GABA release from interneurons. All right. Thank you for enduring my presentation, and have a good day. See you.